Okay, we are live. Um, Good night, everybody. Good afternoon for Dr. Guan. Today, our lecture will be about microbiome, especially in calves, and will be by Dr. Leluo Guan. Leluo Guan speaking briefly about. She is graduated from Wuhan University in China, and she got her master's and PhD in Kyoto University, that's in Japan. Today, her research is focused on the field of host biome interaction and functional genomics in food producing animals, especially cattle. So, Dr. Wan, you can start.
Okay, thank you, Stella, for your nice introduction. And also thank you for Diego's invite. It's my great pleasure to be here to share with some of the research in the microbiome research, especially in the calf area. Let me go back. So, today I'll focus on how we can potentially use microbiome solution and potentially to develop strategies to improve neonatal calf health and performance. So in addition to the brief introduction by Stella, just give you some ideas what I do. So my research area mainly focus on the interaction between the cattle and gut microbiome. Gut microbiome here refer to the whole gastrointestinal tract, including rumen as well as the lower gut. So my lab has developed metagenomics, metatranscriptomics based approach to understand not only which organisms are there, but also what they do that can potentially affect their metabolism, their metabolites, and subsequently affect cattle's performance. So in the past, we have been working on how the microbiome can affect feed efficiency, methane emission, as well as the cattle health. And also, as part of my research expertise, I work on functional genomics. So we have developed a method to study the overall transcriptome, proteome, metabolome, and their regulatory mechanisms in cattle. So specifically in past of 18 years, so what I've been working on how potentially genetics, nutrition, and also the variation in cattle performance can be associated with gut and its microbiome, and how those potentially relate to or associate with animal health and welfare. So you can see here there are three categories here. So my research program was mainly interested in the gut and its microbiome in feed digestion and nutrient absorption, and gut physical development and maintenance, and also how they can potentially inhibit or prevent foodborne pathogen colonization, as well as how the gut microbiome affects host immune system, its development and regulation. So in addition to the traits I mentioned in the previous slide, we actually also trying to understand whether rumen and lower gut microbiome can potentially affect the milk production, including the milk yield and quality, and as well as a meat quality. So we also working on some disease resistance, immune function especially in the early life, and many other traits. So this is basically what I do in the past 18 years. But today I will only focus on the research or the conceptual understanding of early life microbiome in neonatal calves and how much we understand them and how we can use those information to improve early life's health, but also long-term productivity. So before I start with talking about microbiome, I want to give you some definition as well as the facts of the gut microbiome. Okay, so you can see here, which is really important. So the gut microbiota usually has multiple kingdoms, including both eukaryotes and prokaryotes, plus viruses. So this is, very simple kind of a figure in the left. You can see here, I listed the prokaryotes, which includes bacteria, archaea, and eukaryotes, fungi, protozoa, and the viruses. So they are all members of a microbiome and or a microbiota. So you can see here, many research have been mainly focused on bacteria, especially in the lower gut. And recent study has started to review the archaea in the lower gut and also fungi, as well as parasites. Specifically viruses, they can play a very important role in affecting the structure and the function of our overall microbiome. Of course, in the rumen, this have been well studied. We all know in the rumen of cattle, bacteria, archaea, fungi, protozoa, they actually interact with each other. They work systemically and play a role in fiber digestion, VFA production, and so on. So the second key point is gut microbiota is region dependent. And we see lots of a human paper, they use fecal samples to indicate what's going on in the gut, which is incorrect. Lots of research has showing now, especially in the animal system, the microbiota in the small intestine is different from the large intestine, which is also different from rectum and or feces. So when we use the fecal samples as a example or samples to study gut microbiota, we need to interpret the results very carefully. The third part is within the gut, there are two components. We usually call the digesta versus the tissue. So the lumen content associated microbiota actually is different from mucosal attached microbiota. And most of the research only focus on lumen or digesta associated microbiota. And there is a very, very limited study to understand the mucosal attached microbiota because it's not very easy to get samples, especially from alive animals. However, in the real situation within the animal system, mucosal attached microbiota, they actually directly interact with host, especially for some of them attached to the mucin layer, and some of them invade mucus, and they can reach to the epithelia. They can have direct interaction with the host. So this is actually really important perspective. So if you want to design some research in mucosa attached to look at host microbiome in cattle or in pigs, in other animal species, it's important for you to think about whether you can incorporate those mucosal attached microbiota into your study design. So in the past years, in addition to microbiota play a very important role in nutrient digestion, like carbohydrate, digestion, as well as produce short-chain fatty acid. which can provide energy resource for host animals. Past research also shows gut microbiota can affect host metabolism directly. So I list a few research areas, from most of them from human research. It indicates gut microbiota and its metabolites can regulate gut hormone release. So that means somehow depends on the composition of the microbiota, the host animal may shift their production or releasing of the hormone, which can regulate many biological processes. Secondly, the microbiota and its metabolites can also modulate the immune signaling, and which can activate or suppress the immune cascade that can affect cytokine chemokine production, but also the cytokine can potentially affect other organ or tissues function, such as liver and brain and so on. They also lots of evidence showing that gut microbiota, especially in the small intestine, can affect the bile acid metabolism. So some microbes can convert primary bile acid to secondary bile acid, which can affect a growth hormone, which is a GLP-1, that can potentially affect the animal's growth. There is also evidence showing gut microbes can regulate lipid absorption in the gut, especially in the duodenum and jejunum, which also showing in early life in the human, if the gut microbiota had the dysbiosis, which can impact on lipid absorption, subsequently lead to adult obesity and so on. Lastly, as I mentioned briefly above, microbiota and its metabolites can also affect the liver function, which I will not touch about this, but I want to give you some kind of advances to date. What are the knowledges we know so far about how the microbiota can affect host metabolism? So I'll start to talking about early life gut microbiota, or early life gut microbiome, and we have been working on in the past many years. So if you are interested in the early life microbiome, if you do literature, there's a lot of research in human, and they look at infants, at birth, and they talking about the natural birth versus C-section. They're also talking about the breastfeeding versus formula feeding and so on, which can all affect the early life, gut microbiome. So you can see here, after animals are born, or, like in human, you can see there is a graduate establishment of the gut microbiota. So microbiota will have this process like initial colonization, and then depends on how they were fed and how the environment changed. So the establishment of the microbiota can vary among individuals, as well as also, even the same individual between different stage. So in humans, now it's highly recognized the gut microbiota will be established, or they also use the words mature, at about 2 to 3 years old. And then if you look at calves, so this is the review done by my previous PhD student, who actually was the first to working on the early life microbiota of the calves. And we actually did thorough literature review, and also based on our own data, we found likely the early life microbiota of calves usually establish when they become weaned, because now they start to have a solid food intake, they can actually adapt to the high concentrate diet and a starter. So their microbiota in the gut can be established at the time. So regardless, you can say here that usually we know early life gut microbiota is highly dynamic, and also their pioneer microbiome can also differ among different individuals. I'll explain the pioneer microbiome a little bit. So the pioneer microbiome refers to the microbes that firstly colonize the gut. So they usually are the organisms, they colonize the gut as the first ones. So in the humans, we already know certain organisms are the pioneer colonizers. So the pioneer colonizers usually are bifidobacteria. I'll talk about bifidobacteria a little bit later. And gut microbiota establishment or maturation will depend on pioneer colonizer, but also microbial succession. So what does microbial succession mean? Microbial succession means the order of the colonization. So who come first, and then who come next, and then who come to the third. And then this process will then determine the metabolic core microbiome, which means microbiota can change day by day, even different times through the day, because there is different environment organisms coming, there is a diet coming. However, there is a group of core microbiota within the individual. They perform the core metabolic function, which is allow them to provide substrate, to interact with host, to train the host in terms of their immune function, as well as to prevent pathogen colonization. This process also affects microbial interactions. So microbial interactions in the gut means they will recognize which microbes coming from the environment as buddies, friends, or which ones are enemies, or which ones just, they're okay. They'll just be there. I'm not going to work with them, or I'm not going to be friends with them. So microbial interactions actually play a very important role in regulating or affecting the microbiota and its function. So this is why now there are lots of research ongoing trying to understand microbial interactions in the gut. So those microbial interactions could also affect the growth rate of the microbes, as well as the products they produce, which can subsequently affect the baby's gut health, and also, potential baby's health and disease resilience, and so on. So you can see here, research does show that microbial metabolic maturation is highly correlated with host immune maturation. So this means the microbiota maturation process plays a virtual role in affecting host immune function development. And that's why these days early life microbiome play extremely important role in affecting lifelong health. So that means the early life microbiome maturation affecting host immune function development maturation, which they actually directly impact adult human or adult animals' productivity and health. So this is actually also really interesting understanding of how the early life microbiota can affect the host metabolism. So in addition, people saying they also looking at how the early life microbial perturbation can lead to metabolic dysfunction. So microbial perturbation can be caused by disease, for example, diarrhea, or medicine, drugs usage, like antibiotics, or stress. So potentially animal had transportation stress and so on. So all this can potentially cause the dysbiosis of the gut microbiota, which lead to metabolic dysfunction. So here listed a few examples. So here listed organisms called the bile salt hydrolase producing bacteria. So BSH represents bile salt hydrolase producing bacteria. So what those bacteria can do here, for example, the bifidobacterium species and bacteroid species, they possess the enzyme called bile salt hydrolase. So they can convert the primary bile acid to secondary bile acid, as I mentioned before. So when the secondary bile acid pathway are affected, you can see here, they can affect host energy harvest and fat storage. They can also affect immune homeostasis and inflammation. They can also affect the gut barrier function. Similar here, you can see here, HMO means human milk oligosaccharide utilization bacteria or utilizing bacteria. So human milk oligosaccharide, you can adopt this to the cow as well. Those bacteria, they produce short-chain fatty acids. Many include acetate, propionate, butyrate, and they can have all sorts of functions here. You can see there's 5 arrows. Short-chain fatty acid can affect the satiety control, lipid and glucose metabolism, energy harvest and fat storage, also immune function, gut barrier, and so on. Of course, the gram-negative bacteria, usually we know they produce lipopolysaccharide, LPS, and we usually consider them are opportunistic pathogenic organism or the pathogenic organism. And we know that LPS are directly related to the diabetes, metabolic dysfunction in human, because it can affect the lipid and glucose metabolism. It can also affect immune function and the gut barrier function. So this gives you some examples how the microbes can potentially affect their metabolites, and their metabolites can affect the host metabolic function, which can directly affect animals' productivity as well as the health. So I'm going to give you some kind of research outcomes we did in the past years. So how the early life gut microbiome and gut development and functions in the neonatal dairy calves. So in the past, we have been working on we had a question when do microbes colonize? So we performed a study, which is a long time ago, which is also by Dr. Mel Mutegay. So we actually collect calves 5 minutes after birth. So all my students are actually staying in the farm, and we take turn to observe which cow will start to give birthing, and we predict that cow will produce a male calf. So once we got calf at birth, we actually carry them in the laboratory, and then we actually process the calves, collect samples, and looking at microbiota. So you can see here, surprisingly, at birth, we found very, very high diverse microbes in the rumen. even though their abundance was pretty low. Same here, we also found very high diverse microbes in the small intestine. Here is ileum, distal jejunum, and a proxy jejunum. And we also look at digesta-associated microbes, as well as we call the epimural microbes, which is attached to the mucosa. So this gives us the idea is we don't know yet whether the microbes colonize in the fetus. Now there are two groups of kind of researchers. One group of researchers believe there is a microbes already colonized in the gut of the fetus, but majority of the researchers believe the colonization may start from the birth canal, and then at the birth we are able to detect them. So there is a debating, and there is a quite some papers, in Nature or in Science. They talking about the fetal microbiota. I think for some of you are interested, you can look at those, and also you can also form your own idea. Whether there is a microbes colonize the fetus. So personally, I believe there is a very, very, well, I would say fetus is sterile, and because that's where the germ-free animals are derived. However, there is a detection method using this molecular technology could be potentially a background that lead to some of the unconfirmed findings, whether there are some microbes in the fetus. So this is also what we did. I just want to give you some idea what I meant. The gut microbiota differ among different gut regions and also between the contents and the mucosa attached tissue. So this is the data from the calves, and you can see here the small intestine microbiota composition significantly different from the cecum and the colon and the rumen. And you can see here those regions are more similar, but they were still significant different. So the rumen is different from the hindgut, although they both have the function in terms of fermentation, and the small intestine are actually quite different from other regions. And you can see here the tissue attached also significant different from the contents. So this is the experiment design. You can see here what we did, and you can see here we then collect samples at birth and also at day 7, and then 21 and 42 just after the weaning, and we collect whole gastrointestinal tract. We look at the RNA of the tissue, we look at the DNA and RNA of the contents, and we did transcriptome analysis as well as we did microbiota analysis. So we try to understand how the early life microbiome established in the calf and how potentially those established microbiota can affect the tissue function, which we used RNA sequencing because we can profiling the whole transcriptome of the tissue. So this is what is interesting is, so you can see here, we actually identified very different kind of a population of the microbes of the colonization. The first question is we want to see whether the DNA-based analysis is representative. So you can see here, the DNA-based analysis can overestimate alive animals, alive bacteria, So you can see here, the blue bar shows the total copy number based on RNA evaluation. And you can see here, there is a huge difference between the DNA and RNA-based analysis. And also you can see here that the function of the microbiome, so I presented the diversity in the previous slide, and you can see here, based on the metagenome, we identified there is a very diverse or active function of the microbiome through the early life from day 0 to 6 weeks. So they have very, very active already carbohydrate metabolism, protein metabolism, of course, self proliferation, and so on. So what is interesting, in addition to look at bacteria, we also look at the archaea in the rumen. So here is we're talking about the rumen because we all are interested in methane mitigation. And what is interesting, we couldn't detect archaea at birth. Okay, so some researchers saying, oh, archaea also colonize at birth. Actually, we couldn't detect archaea at birth at all. And you can see here, even at 1 week of age, the archaea population is not very high. And similar like bacteria, they gradually increase their population, and the population are pretty established at 3 weeks of age when they started to have a starter. And you can see here, archaea composition is simpler compared to the bacteria, and we identify the main archaea families and the genera and in the rumen based on the metagenomics. So when you look at the small intestine, so you can see here, the microbiota also gradually become more established from 1 week to 3 weeks to 6 weeks. So what is interesting though, we were able to detect bacteria population in the digester, but we actually couldn't get bacterial profiling from the animals at birth. So somehow we were only able to detect microbial profiles attached to the tissue and from 1 week of age. So when we look at calf microbiota, so another extreme important information we got is the microbiota is highly diverse, but we found there is a high individual variation. So this is actually really extremely important. So if you look at those dots, okay, each dot represents one animal, and you can see here, they were not really necessarily very similar. They were actually highly variable. So some individuals have way more higher firmicutes, but some have very, very low firmicutes. So there's a huge variation among individuals. So this is actually really important, okay? So for many nutritional study, if you want to treat a group of cows with feed supplements and you want to look at microbiota difference, it's extremely important for you to take the individual variation in microbial composition into consideration. So how you can do that? You need to collect the baseline samples before the treatment starts. So later, we can actually include this individual variation as a covariance in the model, so we can determine whether the observed microbiota difference is due to individual variation or due to nutritional treatment. So similar here, not all the calves are born well colonized with the same microbiota, even though they will be raised in the same farm fed same colostrum. So the reason is because they will come from different mom, right? There will be different environment during the birth process, the birth canal. It also could due to their genetics, because their genetic component will select different microbes to colonize. So another very interesting findings we found, which is actually form the base for us now to isolate probiotics, is you can see here when calves are born, in their gut, the bifidobacterium, we know bifidobacterium are very important organisms. They are actually very dominant in the small intestine. And you can see here, there are very, very high proportion, like here from 10% reached to 20% in the first week or 3 week, and they still maintain as a 59% of the total population in the small intestine of the calves. So we know bifidobacterium are very important because bifidobacteria are one of the groups of the probiotics, and it has been widely studied in the human. And you can see here, bifidobacteria, they can produce the exopolysaccharide, so make them can form biofilm along the epithelia. So they actually can potentially pass through the mucus. They can actually form the biofilm, and the EPS of the bifidobacterium, they can modulate the immune system of the host. Because they can bind the mucus, epithelia, they actually can prevent pathogens to bind or to invade the host epithelial tissue. And also what is interesting, bifidobacterium form the biofilm, they can actually scavenge the radical oxygen species and also toxins, as well as the potential toxic compounds to the gut microbiota or to the host, potentially affecting or negatively affect host microbiome interactions. And what is interesting, once the bifidobacterium form the biofilm will not just be potentially bifidobacterium, they can also modulate the microbes, they can pass through the mucus, they can attach to the epithelia. So this is why now, if you want to really look at bifidobacterium, which in human study considered bifidobacteria are the pioneer colonizers, and why they were so important, and also why we want to boost bifidobacteria during the early life, especially in the neonatal animals or neonatal infants in the human. So bifidobacterium also play very important role in affecting the host function. Here is also one of my another previous PhD student who is now a professor in China, who actually identified the bifidobacterium and lactobacilli. Their abundance can potentially relate to the expression of the microRNA, which are the regulators of the host gene expression. So you can see here, bifidobacterium lactobacilli could play very important role in crosstalk to the host animals and affecting the expression of genes that contribute to the gut tissue development and also the animal health. So I didn't include, but we actually identified direct interaction between the microRNA and microbes in the gut, together with the microRNA interact with the host transcripts. So through the study, we also found, when you look at the calves, when they're talking about the individual variation, we found some calves, they actually have a lactobacilli dominant in their small intestine, and some of them have a bacteria is dominant in their intestine. But what is interesting, you can see here, in the gut of lactobacillus dominant calves, they have varied different expression of the genes, and those genes are mainly involved in immune function. You can see here enriched function in leukocyte and lymphocyte chemotaxis, cytokine mediated signaling, and inflammatory responses. Well, when you look at the bacteria-dominated calves, their small intestine are mainly enriched function in cell adhesion, response to stimulus, cell communication. It's more like cell function, lack of kind of inflammatory response or recognition of the antigens produced by the microbes. So I'm not going to talk about this in details, but this is one of the study we actually found, as I mentioned, how the rumen microbiome interact with host microRNA as well as the tissue messenger RNA, so they can regulate the rumen development. So we published this study in the Genome Biology. Okay, so I think I might take a lot of time, but I will just summarize this section quickly. So there is a lot of factors affecting early life of the microbiome of the dairy cows, including diet, colostrum, milk, and calf starter. Of course, antibiotics, probiotics, age, and host genetics. I know that I only have 15 minutes to talk, but I want to maybe just introduce a little bit more about how the colostrum feeding can affect the gut microbiome and how they shift in neonatal dairy calves. So I just give you some idea that we do microbiota research, but we want to apply those research into the real situation. So this is the research we found. The feeding fresh or heat-treated colostrum, they can actually reduce the E. coli population in the digesta as well as the tissue attached in the small intestine. And same here, you can see in the large intestine. So this is the colon. And also feeding fresh colostrum and heated colostrum can actually increase the proportion of the bifidobacterium. And you can see here, this is alien attached. And this is actually within 6 hours and 12 hours. So we want bifidobacterium to colonize as much as they can within the 24 hours after birth. Same, we found the fresh colostrum and heat-treated colostrum can boost bifidobacterium in the colon attached, colon tissue attached microbiota. So this suggests feeding colostrum is very important. Doesn't matter fresh and heat-treated, it will play very important role to boost the beneficial bacteria, bifidobacterium, reduce the opportunistic pathogen, which is E. coli. But based on our study, you can show here the heat-treated colostrum, it is likely boost bifidobacterium more than the fresh colostrum. So now just give you a message. So feeding colostrum is extremely important to help the pioneer colonizer as well as the establishment of early life microbiota. So this is, I just want to show you, we also look at how the delayed first colostrum feeding can affect microbiota. Apologize if this is too detailed, but you can see here, we also focus on the altered mucosa attached microbiota in the ileum and the colon. Basically, we found the delay colostrum feeding for 12 hours, it can significantly reduce the lactobacilli attached to the mucosa, and also it is potentially are affecting the relationship between the short-chain fatty acid and the mucosa-attached microbiota. So when the colostrum feeding is delayed, it will likely increase the opportunistic pathogens, reduce the short-chain fatty acid. So this is actually really important now. Not only we need to feed colostrum, we also need to feed colostrum timely. So that's why the industry recommendation feeding colostrum within 4 to 6 hours is extremely important. And then we also looking at how the individual calves respond to the delayed colostrum feeding. And we found not all the calves respond in the same way. We found some calves, they actually can be more resilient, even the colostrum is delayed, colostrum feeding is delayed, but some calves, even they fed colostrum on time, they may still not perform well because they have varied initial colonization of the microbiota. So here we identified delayed colostrum feeding can affect the taurine metabolism, especially affect the receptor of the host tissue that can impact on the pathogen recognition. Okay, so basically the understanding is not all the calves have the same similar microbiome, and when we have different colostrum feeding practice, some calves, usually the delayed colostrum feeding will overall affect gut microbiota, but not all the calves will respond to the delayed colostrum feeding in the same way. So I think, which is the same thing, I'm going to pass this. I will send slide to you later if you're interested. Oh, basically on her pass, I would just want to show you here, when we look at individualized microbiome, how they're affecting their resistance or resilience or susceptible to delayed colostrum feeding. You can see, depends on their microbiota. We also observed varied ileum, kind of a length and crypts, but also their insulin measured in the serum. So that means the microbiota play very important role in the small intestine development and potentially the calf metabolism. Okay, so the last story I want to share is we also found the colostrum feeding strategy can affect the expression of the neuroendocrine genes and also active mucosa-attached microbiota in calves. So this experiment design, we actually fed calves with whole milk and half colostrum and half milk. And then we had colostrum feeding. Basically, we extended those feeding practice. So we hypothesized the extended colostrum feeding can improve the gut health by boost the bifidobacterium population, reduce the E. coli population. However, in this study, I don't have time to explain other things, but what I want to share with you is what is interesting we found by changing the feeding practice. We found the expression of endocrine receptors, and their expression pattern significant different while they actually among those different treatment, the feeding treatment. So you can see here different feeding can potentially lead to lower expression of certain receptors. Here the steroidogenic acute regulate protein, which is regulate the steroid production, as well as adrenic receptor, which is also related to the cortisol production, as well as the serotonin receptor and also serotonin transporter. So you can see here, feeding practice can affect the gut microbiota. Gut microbiota can potentially affect the endocrine molecules or hormone secretion. As I mentioned in the human study, we also found evidence in the calf. So this is my, I think, the last slide. Well, one of the last slide is about implications. So now you can see here, research as showing that the manipulate microbiota can potentially improve the pre-weaned growth and also reduce the weaning stress. And also microbiota can be used to modulate their susceptibility to the early life diarrhea and the pneumonia. I don't have time to talk, but there is evidence showing gut microbiota can affect the lung function in calves as well. Another interesting perspective is we can potentially manipulate or intervene rumen microbiome in early life, which can lead to improved feed efficiency and the decrease the methane production during the later life of the cattle. So I think this is the what I want to say, how potentially we can use different strategy to manipulate the gut microbiota through nutrition, management, and the feed additives. So these days, lots of researchers are thinking about how to manipulate microbiome. They can use potentially the direct fat microbes, probiotics, postbiotics, and the psychobiotics. Okay, so I'll pass all the slide. I think I'm over the time, but I want to give you some idea about the new idea about the psychobiotics. So the psychobiotics are the probiotics can potentially affect the gut-brain axis of the calves. Here, potentially can reduce the stress of the calves. So this idea is widely adopted in the human study, but it has not been done in the cattle or ruminant research. So here is the way that we are thinking. Maybe we can identify the probiotics. They can potentially, affect calf or cows intake, average daily gain, and appetite or behavior scores during the stressful time like weaning, calving, transition, and so on. Okay, so that's the last slide. So the take-home message, what I trying to talk about today is there's a diverse microbes that colonize the gut of neonatal calves soon after birth, with the bifidobacterium as pioneer colonizer. Colostrum feeding and management can influence the early life colonizer, prevent pathogen colonization, which can also potentially affect the host gene expression, microRNA expression, as well as endocrine function that lead to animals' behavior and stress responses and so on. So I guess we still need more study to understand the mode of actions of the bovine-derived direct fat microbes or probiotics, the psychobiotics, and so on. So we can potentially make commercialized products and the industry can adopt to improve calves' health and performance. With that, I thank all the collaborators, especially Dr. Michael Steele and Dr. Alan Lahman and all the students, and also visiting scientists from Japan, generating all the data I presented today. And also thank you for your attention. Maybe I organized too much slides. I'm a little bit over time, but thanks a lot. And I'm happy to answer your questions. Thank you, everyone.
So, Dr. Guan, excellent lecture. I want to thank you in name of all students of UFMG and everybody watching. It was really good. I learned a lot. Everybody can send the questions on the chat. I will read. But first, I have a question of my own, that if that's okay.
Should I stop sharing slide or should I keep it on?
No, there is no need. I can put it back or take it off.
Okay, no, I need to from my screen so I can see you. And
That's okay. You can stop.
okay. Okay. Yeah. Now I can see from my desktop.
Yes. So my question is about what is your opinion on the relationship between the rumen protozoa and the bacteria present in the rumen, and like how the protozoa potentially can act as a regulators of the ruminal microbiota or perform other functions as well.
Yeah, I think for protozoa's function is still not really well understood. So some researcher, well, based on literature, it says protozoa usually will engulf the bacteria. That's most of the cases there. That's how they get feed. But protozoa can potentially directly involved in carbohydrate metabolism, especially starch degradation, and also they can potentially involved in urea recycling, ammonia recycling. So in this nitrogen kind of a metabolism. But what is the challenging is we know that dephonation. So there are study people actually dephonate rumen. So dephonation actually will remove protozoa. So people use the chemical, or people used, you can actually take out all the rumen contents, leave them under the air for 10 to 15 minutes, the protozoa will die. And when they put back to the rumen, so this is called the dephonation. And in dephonation, they can also add chemicals. But what happens is, some study shows dephonation affected cow's performance. Some study shows there is no effect. So we don't know whether protozoa is really needed, because half of the study show there's no effect. Half study show there is effect. I guess that depends on what they eat. Secondly is protozoa is very, very, very difficult to grow. So we can study them as a pure in vitro, because they always comes with bacteria and a methanogen. So if you want to isolate protozoa, there's always bacteria archaea in the protozoa cell. So it's very difficult to study protozoa as a pure organism. So there's a few studies recently focused on protozoa, and they try to emphasize protozoa are important. However, the protozoa classification still challenging, and their functions still are quite predictive, because we cannot really isolate protozoa separately. But people do believe, though, 30% of methanogen archaea are associated with protozoa.
Yeah, okay. Thank you very much. The next question is
First, Stella, if I may, I just would like to thank you, Dr. Leluo, very much for accepting our invitation and to take a time and share your experience with us. And we know that you have been publishing and studying the cattle microbiome there since a few years now, right? And it is great to see how we develop this field of studies, and how your studies specifically were very important. So for me, I normally work more on the field than in the lab, and I have very few experience with microbiome. And my question is uh, about probiotics, and how in which step in probiotics development we are, like, do we have much evidence of their effect, and when to use it, when is beneficial and we have a good effect, and when does not have effect?
Yeah, I think it's a really good question. When I was in Florida in ADSA, compared to past years, there are lots of probiotics right now. Different companies say, and there's different trials. They test different companies, products. So for the probiotics, based on the literature, there's still limited effective products because most of the probiotics, they are not derived from cattle. Just like, we use probiotics, many of them are actually from milk fermentation or food fermentation or human probiotics. So this is actually a large issue on probiotics is, for example, if we give humans probiotics to fish, actually it may cause fish die, which is really interesting recent finding. So that means we don't have a calf-derived probiotic or cattle-derived. Secondly is for the probiotics, it will be very difficult if we, let's say, Lactobacilli bifidobacterium for adult cattle, because we want Lactobacilli bifidobacterium target hindgut. So it can only used for calves and when rumen is not developed. So if you want to target a rumen microbiota using probiotics, which is most of the product right now are yeast and yeast fermented products, that will be only target adult animals. But still, yeast may not be very functional because rumen is obligate anaerobic. Yeast are aerobic organisms. So that's why we still have a lot of unknown kind of factors. That's why if you do meta-analysis or probiotic research, some people say there is effect, some people say don't. So the overall, it's very not conclusive.
Okay, thank you. And when we should use it, like for example, thinking about calf diarrhea, should we start to use it just in the early life or when the diarrhea starts?
For diarrhea, I would say first week of life, because that's where diarrhea usually happen after first week of life until third week or fourth week of life, right? So that's why we actually feed calves with probiotics, bifidobacterium, first week of life.
All right. Okay, thank you. I let you, Stella, continue, please.
Okay. There's a lot of questions. So
Really? I can't really see.
yes, I
Scare
will put
me.
on the screen so you can see it. So first one is from Isabella. See you now, you can see it.
Okay,
But it's in
cool.
Portuguese.
I like your technology now. Okay.
Yeah, she's asking. She first is given congratulations. She loves the lecture, and
Thank you,
she's
Isabella.
asking about the bifidobacteria, if they influence on the weight gain of the calves. If the calves, you understand? My English is a little broken.
Yeah, so that's a really good question. Bifidobacterium themselves may not directly affect calf weight gain. However, bifidobacterium have interaction with other organisms like Clostridium. So they have a crosstalk. So what does Clostridium do? Clostridium will produce butyrate. And the butyrate will stimulate the growth of colonic cells, and the butyrate can be utilized for energy resource for the calf. So if the bifidobacterium, if the calves have high abundance of bifidobacterium and Clostridium, so they will likely work together well when they ferment the carbohydrate in the colon. So potentially they provide more energy. The butyrate, when the rumen is not developed, likely contribute to the calf growth gain. I hope that makes sense.
Yeah, I believe I believe it answered her question. So the next question is from Ana Julia. She also liked a lot of the. She's in English. You can read.
Oh, thank you.
So her question is this one.
Okay. Yeah, thank you. Fecal transplant is very popular. I can share with you. I just came back from China last week, and there's a lot of fecal transplant experiment going on in China right now. So fecal transplant is kind of there is a Nature communication paper done by a Korean group. They claim that giving the fecal transplant to calves during early life somehow increase the average daily gain for adult animals. But what is the issue is how you can create a standardized fecal, like the donor fecal material, so they can be standardized, they can be the same, for all the calves you are going to treat. Secondly is fecal transplant feces may also carry some of potential pathogens. So they may not pathogens to some calves, but as I said, calves have individualized the microbiome. So when the fecal microbiota come in, they may consider those microbes as enemy. So they can actually potentially start to react, which can cause the host anti-inflammatory responses or activation or overactivate immune function of the animals. So it really depends. There is no evidence yet in the calves fecal transplant can be effective for diarrhea. I guess for human, there is a product right now in US approved by FDA that they can actually use the capsule to cure the Crohn's disease as well as the IBD. But we need to be very careful because there is no evidence yet that fecal transplant can last very long. So people only report some cases are effective and long, but there's also reports showing even we give the fecal transplant, the recipient's microbiota returned to before. So just like we do the rumen transplant. So I did a rumen transplant in 2014-15. We didn't do fecal, but we did a rumen transplant. We exchanged the rumen between two group of 10 animals, total 20. What is interesting? Some cows' rumen returned within a day, and some returned within 7 days, some returned with 14 days. Some cows' rumen become intermediate microbiota. Some cows' microbiota change completely. So what I want to say is that individualized microbiota and the host genetics, host gut environment can be very important role to affect the efficacy of the fecal transplants.
Okay. Next question is from João. I will put here. It's in Portuguese. Oh, Ana said thank you. João. Okay, João is asking about one of your publications, your recent publications, the one about
rumen. I can see ruminal.
yeah, about, I believe I could say resistant.
Oh, the resistant. Okay, yeah.
Yes, he's asking about how can the diversity of the resistant in the rumen microbiome influence in the rumen's response to different diets or management practice, especially those that use antimicrobials.
Yeah, this is a really good question, Joel. So maybe I'll step back a little bit, give you some ideas. So all the microbes carry resistome. So, well, I would say resistome. I would say not all. Many bacteria carry antimicrobial resistant genes. The reason is microbes. So microbes produce all sorts of antibiotics, antimicrobial compounds. So within the microbiome, to be able to survive, microbes need to produce those antimicrobial or antibiotic resistant mechanisms so they can survive. So one of our recent paper found, if you look at the genes, which is resistome of the rumen microbiome, you always identify all sorts of, resistome, the genes, because there are so many bacteria there. There's so many of them carry those genes because they need to survive. But depends on the diet. Not all these genes are expressed. So what that means? When those genes are not expressed, that means microbes are not necessarily resistant to those antibiotics. So we found that Prevotella, there are more resistance. The expression of their antimicrobial resistant genes are highly expressed in the high-grain diet because Prevotella, they need to survive, and they want, because they play a very important role in starch degradation and fermentation. So my hypothesis is bacteria, some function like carbohydrate degradation and metabolism, it's depend on their survival. So antimicrobial resistance can be very important for bacteria to survive, but then also perform a function. So in the rumen, antimicrobial resistance may not necessarily be negative if they don't cause the human pathogenesis or they cause the cow disease, because they only use for fermentation or fiber degradation. So we need to look at antimicrobial resistance in a different way. So for pathogens, if they cause animal disease or foodborne pathogens, they can transmit to human, that's a problem. But if they are resistant genes in the organisms, they are not pathogenic, they are doing fermentation, we need to consider it to be more functional and potentially could be beneficial to the animals in terms of digestive efficiency. I hope that helps.
I believe it does. So that is all the questions. And that's it. I would like to thank you again, Dr. Guan. It was amazing. I learned a lot. I believe everybody have learned. And thank you very much.
Yeah, thank you. And if you want to learn rumen microbiome about cattle, I can share with you sometime again. So it's my pleasure, Tiago. Thank you so much to invite me and to share our research. Apologize, I rushed a little bit in the end.
That's okay.
But thank you so much.
Yeah. Thank you once again, Leluo, and don't need to apologize. Take the time as you need, and thank you for sharing all this. I have some new informations I haven't heard about, like the psychobiotics, for example, and it is a really interesting thing. So yeah, I hope we have some time and opportunity to share again this the opportunity and the research. Okay, and so for this I say goodbye and thank you, Leluo.
Yeah, thank you. If your students or if you or other ones want, I'm happy to share the PDF file as well.
All right, thank you.
Okay.
Okay. Thanks again. Nice to see you both, and also nice to see everyone. Bye.
Yeah,
Goodbye.
bye.
Now I need to know how to get out of. I guess you can start.